Communication is important to both the individual and species level. In addition, integration of speech is important in establishing relationships as well as oneself and society. The FOXP2 gene is a vital component of speech acquisition and language in humans, as well as vocal production in birds and mammals. Rodents communicate through ultrasonic vocalizations, or USVs. The process of ultrasonic vocalization recording in the pups is shown above.
This image shows the location of the FOXP2 gene in the genome.
FOXP1 was quantified in this experiment using Western blot analysis.
As an overview, the male rat pups had a more successful call than the female pups because they were brought back to the nest more often than the females. This could be attributed to the fact that the male calls were found to emit USVs at a lower frequency and amplitude than the female rat pups, as well as the fact that male rat pups have a higher amount of the FOXP2 protein. Due to these reasons, the FOXP2 protein could be a major factor behind the differences in vocal communication between genders. After gathering the results from the rat pups, this experiment was then furthered to be related to humans. With humans, the opposite was found to be true. This means that after examining the cortex tissue of both 4-year-old males and females, the amount of FOXP2 protein in females was found to be higher than of that found in males.
The reason we conducted this research is to gain a further understanding of whether there are identifiable sex differences in rat-pup ultrasonic distress calls and what determines their sex difference. Additionally, we would like to determine whether the mother, which is called the dam, perceives a sex difference, thus altering her behavior accordingly. Birdsong is important because of the significant sex discrepancies in some species, resulting in the discovery of the first sexual dimorphic neuroanatomical underpinnings regulating indefinite behavior. FOXP2 is a member of the 4-cut family of transcription factors and has a highly conserved amino acid and DNA sequence, as well as a conserved distribution in the brain across a range of species that exhibit some sort of vocal communication. It regulates the expression of a large number of genes associated with brain patterning, neurite outgrowth, axonal morphology, and dendritic branching. Mutations of this protein in the humans manifest in speech impairment such as stuttering. Upon studying the animal kingdom, it becomes evident that there are significant differences in auditory communication across genders. Although prior to this experiment, there existed no evidence of male versus female signaling variations.
The materials that were used in order to conduct this experiment were pups born from pregnant Sprague Dawley rats from Harlan Labs, food and water, frozen human cortex samples taken from deceased male and female children, control siRNA, RNA from frozen human brain samples, and staining made from nickel diaminobenzidine.
Methods. Pups were delivered from the pregnant rats under normal standard lab procedure. PN0 was when the pups were found in their nest, and this was considered to be the pup's day of birth. The pups were given food and water ad libitum. Pups were then isolated from maternal interaction, and vocal emissions of 4-day-old pups were recorded for 5 minutes each. Background noise was eliminated by high-pass impulse response filter. Parameters: fundamental mean frequency, fundamental mean amplitude, frequency bandwidth, duration, and number of calls emitted. Methods continued. RNA was prepared from frozen human brain tissue. Immunohistory was used by rinsing tissue with PBS and polyclonal antibody against FOXP2 and incubated. FOXP2 positive cells were detected by using nickel diaminobenzidine in order to stain FOXP2. This was used in order to compare amounts of FOXP2 in rat pups and in humans.
Dams retrieve males before females because of their USVs. The dam retrieved the pups as shown in panel 2A. The rank order of pup retrieval based on sex in controlled dams with litters that received no treatment indicated male pups were more likely to be retrieved before females. When litters were treated with either siRNA against FOXP2 or scrambled control males were more likely to be retrieved first and females retrieved last. This visual shows that FOXP2 is differentially expressed in boys versus girls' cortex. The relative amount of FOXP2 and FOXP1 protein was quantified by the Western blot analysis. The cortex from girls had significantly more FOXP2 than the cortex from boys. In these same samples, there was no sex difference in level of FOXP1 protein. mRNA levels for the FOXP2 gene showed a significant positive linear relationship.
The results produced were statistically significant when comparing male and females. The male and female rats were killed on PN4, and regions of the brain were observed to include the cerebellum, amygdala, cortex, thalamus, hypothalamus, hippocampus, and preoptic area, or POA, using Western blot in order to understand the FOXP2 protein. Males had significantly higher basal levels of FOXP2 protein than did females in the cerebellum. No sex variations in FOXP2 levels in the hypothalamus or POA were found. No FOXP2 protein was found in the hippocampus for either sex. In conclusion, sex variation is a result of generalized reduction in FOXP2 protein in each cell for males in comparison to female, with this reduction not being significant enough to alter the detection of FOXP2 within individual cells through means of immunohistochemistry quantification.
This image shows siRNA directed against FOXP2 mRNA and reduced FOXP2 protein levels in both the cortex and striatum C. By postnatal day 4, there was a prevalent sex difference, FOXP2 protein levels with males having higher levels than females, and the suppression of FOXP2 levels by siRNA was no longer apparent. However, females treated with FOXP2 siRNA showed an increase in FOXP2 protein compared with scrambled control-treated females, suggesting a rebound following inhibition.
As displayed on this graph, male pups have higher levels of FOXP2 proteins in each region of the brain except for the hypothalamus and POA, which are regions of the brain that are not associated with vocalization and communication.
In order to determine their identifiable sex differences in rat pup ultrasonic distress calls, we examine the ultrasonic calls of individual 4-day-old pups separated from the dam for 5 minutes. Isolated pups made hundreds of calls during separation, with male pups averaging almost twice as many calls as females. We further sought to determine the effect of treatment with an siRNA directed against FOXP2 mRNA on pup USVs. Two-way ANOVA showed a significant correlation between treatment and sex. Control males had 603 vocalizations in comparison to 350 produced by control females, whereas siRNA-treated males produced 466 total vocalizations, which was fewer than controlled males. Additionally, siRNA-treated females produced 465 total vocalizations, which was more than control females. This was not a hypothesized outcome. We analyzed the fundamental mean frequency of vocalization. Controlled males vocalized at a lower frequency than control females, and treatment with siRNA reversed this pattern so that males treated with siRNA against FOXP2 made calls that were higher in frequency than control males. Surprisingly, females treated with siRNA made calls that were lower in frequency than control females. Male pup ultrasonic vocalizations are different from female pup vocalizations, and FOXP2 siRNA reverses this pattern. Infusion with siRNA or scramble control RNA to 4-day-old pups resulted in control-treated males making almost twice as many distress USVs per minute time as control-treated females. The mean frequency and amplitude of the calls are significantly lower in males compared with their female litter mates. Treatment with siRNA directed against FOXP2 mRNA 2 days before testing significantly reduced the total number of calls in male, but increased the number in females. The same pattern was seen for frequency, whereas amplitude was modified by siRNA treatment only in females. We analyzed the mean amplitude of each call produced. Control males vocalized at the lower amplitude than control females, however, siRNA treatment reversed in this pattern, but only in females. We also analyzed the control group that did not receive any injections. Upon further evaluation of the siRNA at reducing protein levels of FOXP2 levels in neurons, we cultured primary cells from PN0 rat pups using tissue taken from the striatum and cortex. This showed the siRNA was useful in the reducing FOXP2 protein levels in the striatum as well as the cortex. We then attempted to determine the duration of the siRNA suppression of FOXP2. Once again, we saw a sex variation such that the control males had higher FOXP2 levels than males. However, there was no longer a difference between siRNA and scramble control injected females. Control males were retrieved before females, whereas siRNA treated females has a faster retrieval rate than that of the control female group.
We retrieved human cortex results from non-disease donors. All cortexes were from the left hemisphere. The data we found from humans was surprising because it was opposite to what was found in the rat pups. Unlike female rat pups, female children had higher levels of the FOXP2 protein than male children. When the FOXP1 gene was examined, there was no difference in amounts between male and female children. The sex differences were only clearly apparent in areas of the brain that are involved with higher cognitive function. Sex differences are innate, so naturally the USVs that are emitted, although they differ between gender, are unlearned. It was also found that in the control group, males had higher vocalizations with a lower frequency and amplitude than the control females. Another result that was found is that siRNA has an opposite effect in females, but it is unknown the reasoning for this occurrence. In almost all instances, the male calls were preferred by the mother, except when siRNA was present. Another finding was that the human females data is the exact opposite to that of the rat females in the sense of higher FOXP2 concentration than in the male. Overall, we believe that the calls have evolved in order for the animal to have a better survival rate. The hypothesis is never stated throughout the research, as well as there is not sufficient explanation as to why the results were opposite in rats and humans, which makes it more difficult to understand how the research relates to humans. The condition that the rat pups raised in was not specified, as well as there was not enough information provided on FOXP1 gene.